Today we're going to be discussing a little bit about unlocking nitrification and removing ammonia within your respective treatment plants. My name is Michael Frett. I'm an aquatic biologist here at Aquafix. So I just want to say thank you again to everyone for coming here today and joining for today's webinar on nitrification and removing ammonia. At Aquafix, we really have conducted an abundance of research and testing on nitrifying bacteria, both within our lab as well as in the field at various wastewater treatment facilities. And as an aquatic biologist here at Aquafix myself, it's really been my job to research the science behind our treatment system solutions. And specifically, that's going to be based on improving nitrification and recovering nitrification. And I want to share a little bit of that information and those solutions with all of you today. So I first wanted to start this presentation on why we believe this webinar is going to be important and relevant. Well, I think I can reasonably assume that if you've joined us today, each of you have some sort of interest in ammonia removal in your respective water treatment facilities. One of our primary goals at Aquafix is to help assist and truly be a partner for wastewater treatment facilities to achieve the goals of efficient ammonia removal and efficient recovery from elevated effluent ammonia levels. Really in order to mitigate those negative downstream effects that can come with elevated effluent ammonia and nitrogen levels. And so today we're going to be discussing those two goals, as well as covering the following topics. That's starting with discussing nitrogen in general, including the nitrogen cycle and nitrifying bacteria. We're going to take a look at the environmental parameters and their impact on nitrification. And we're also going to take a look at Aquafix's nitrification product, VitaStim Dynamic Duo, and some of the field research as well as in laboratory research that we've done on this product that we want to share with you today. Most of that research, also the production of our nitrifiers and an analysis of our testing, is going to be done within Aquafix's research laboratories that are located in the University Research Park in Madison, Wisconsin. Through this research, we've really been able to fundamentally grow our knowledge base on nitrifiers with that research, and we've been able to refine our nitrification solutions within our laboratories to really help out most within the field. And so now really starting off, I want to talk briefly about the nitrogen cycle. And this figure here is really going to somewhat illustrate the many interactions and forms that nitrogen can have throughout the environment. And although the complex interactions shown here are really not entirely relevant for today's specific presentation, it's important to understand that the nitrogen and ammonia that you reduce in your wastewater treatment facility is going to be a part of this larger cycle. Regarding wastewater sources of nitrogen, the most commonly thought of input of nitrogen is the biological nitrogen in plants, animal waste, and soil runoff, primarily from these agricultural sources. And the industrial production of nitrogen, as well as a byproduct, can also act as an input to not only the environment, but subsequently wastewater treatment plants as well. There's also going to be nitrate in groundwater, since nitrate, unlike ammonia, is not going to be bound to soil or clay particles and can move freely through the soil and infiltrate the groundwater and come into your plant. So if your plant has lots of inputs from infiltration in an agriculturally dense area, you may deal with high nitrate coming into your plant. Biological treatment is used to prevent most of this nitrogen and other compounds as well from ending up in our environment and leading to undesirable outcomes such as algal blooms in lakes, fish death, and the prevalence of nitrate or nitrite in drinking water wells. So speaking of nitrogen, we really can organize it into two primary categories, and those are going to be organic and inorganic nitrogen. We'll start off briefly here talking about organic nitrogen, as it's comprised of really nitrogen molecules that are going to be bound in a larger biological molecule that really contains carbon as the backbone of its construction. Within a wastewater treatment plant, organic nitrogen is typically bound to a plant's influent BOD as a whole. In regard to inorganic nitrogen, we can start with ammonia here. And when we're talking about ammonia, we can really either be talking about the ion NH4 plus ammonium or the uncharged NH3. We also then have nitrite and nitrate, which are two different oxidized products of ammonia and contain oxygen molecules attached to that nitrogen. When nitrification occurs, ammonia is going to be oxidized all the way to nitrate, and that nitrate can then be denitrified all the way to nitrogen gas and released back into the atmosphere. We're going to investigate all these forms of nitrogen throughout today's webinar. And so we'll take a little bit more in-depth look at organic nitrogen here, as it really mostly comes into the influence of a wastewater treatment plant in 3 separate forms. Those forms are going to be proteins, amino acids, and urea. Proteins are going to be long chains of amino acids. And so if you look a little bit more closely within that protein molecule, you're going to see it's a chain of amino acids, and you're going to see that the nitrogen is going to be found on the amino group of that amino acid. We have serine here as an example of amino acid, and you see that you have this nitrogen grouping on that amino acid. So that's where the nitrogen is going to come from in these larger proteins and individual chains of amino acids. And then when we talk about urea, it's really going to be the biggest contributor of nitrogen to municipal influent in most cases. So here we have a urea molecule. At the bottom of the screen, you can see the 2 nitrogens attached to a carbon. And what's interesting about urea is that the urease enzyme, which is actually pictured at the top of the screen here, can break down urea, and in that you end up releasing ammonia to within your wastewater treatment plant. So that's where you're bringing in urea into a municipal plant, and you end up getting this free ammonia eventually that ends up needing to be nitrified. So let's take a closer look at that nitrification process specifically. When we're talking about ammonia, the first step of nitrification is going to be ammonia oxidization. So that ammonia oxidization step is going to be ammonia being converted into nitrite, plus a couple of byproducts in there as well. The next step in nitrification is going to be that nitrite then being oxidized all the way to nitrate, as you can see here. Nitrification can be modeled with these spheres where nitrogen is blue, hydrogen is white, and oxygen is red. For plants that aren't nitrifying well, they're not going to be progressing through these 2 separate reaction steps. And either it's your ammonia or it's your nitrite is going to end up remaining within your system and leaving within your effluent. And so as we talk about nitrification, it's important to remember that nitrification is a biological process that takes place with the help of bacteria and the enzymes that those bacteria create. The oxidization of ammonia, that first step to nitrite, was thought to be primarily done through nitrosinomus and nitrospira. We have an image up here in the top right corner of nitrosinomus. This picture is from an electron microscope, as you really won't be seeing these bacteria using a regular light microscope because they simply are too small. But that's what you're looking like when you're talking about these nitrifiers under intense magnification. The next step in the process was then of nitrite being oxidized to nitrate was thought to be primarily done through nitrobacter. As more and more research has come out, nitrifying bacteria really are becoming increasingly better characterized, and there's increasing evidence that nitrosinomus and nitrobacter are not the sole nitrifiers in wastewater and in waste activated sludge, but rather you're going to really find different nitrifying bacteria within these different wastewater conditions, as nitrifying bacteria really tend to specialize to different environmental conditions. So different wastewater plants tend to have really vastly different and diverse populations of nitrifying bacteria as a whole. So as I was talking about those nitrifying bacteria, let's talk a little bit more about nitrifiers in depth. And so nitrifiers are autotrophic organisms, and that's probably the most important thing to remember. So being an autotrophic organisms, it means that they synthesize their energy from inorganic nutrient sources. When we're talking about nitrifiers, this is specifically inorganic nitrogen and inorganic carbon. This process, the oxidization of ammonia, really does not generate much energy comparatively to heterotrophic bacteria. And the utilization of inorganic carbon to gain that cellular biomass is going to really be an energy intensive process. It's not really easy and efficient. And as I said, this is really going to be different than those heterotrophic bacteria that we're used to. and we're going to be discussing those shortly. Some other notes about nitrifying bacteria are that they're really going to be sensitive to changes in environmental conditions, and also they really inhabit a variety of environmental areas, whether that be land, water, soil. Basically everywhere in the world, you're going to find some sort of population of nitrifying bacteria. And so within a healthy wastewater treatment plant, nitrifiers are likely to be present and inhabit flocks of bacteria throughout the mixed liquor. Through these fluorescently tagged microscopic images from a recent scientific paper on nitrification, you can see the communities of nitrifiers as well as other bacteria beginning to develop during the wastewater treatment process. The image on the left, you can see an example of flock at the very initial stages of a wastewater treatment plant's process. However, on the right, we can see a fully developed, healthy flock, as well as a fully developed and healthy community of nitrifying bacteria interacting with each other. You can see the ammonia oxidizing bacteria, such as Nitrosomonas and Nitrospira, in the cyan fluorescent tag, and you can see the nitrite oxidizing bacteria, such as Nitrobacter, in the pink fluorescent tag. One thing to note is that nitrifiers typically inhabit the outer layers of flock, and this really is largely due to oxygen availability and preference. But one drawback about this positioning on the outer edges of flock is going to be the nitrifier's ability to handle adverse environmental conditions, as they're less protected on this outer edges of the flock. Eubacteria in blue would be categorized as our classic heterotrophic flock-forming bacteria within waste-activated sludge, and we will investigate those next. So, as I stated, heterotrophic bacteria are really going to be our other category of wastewater bacteria living within flock. And they really do differ from nitrifiers, as heterotrophs are actually able to synthesize food from organic sources instead of that inorganic nitrogen and inorganic carbon. And so because of this, these organic carbon-based sources of food are really able to generate high amounts of energy in order to support rapid growth of heterotrophic bacteria. Heterotrophs are also going to be the primary reducers of soluble BOD within a wastewater treatment system through the breakdown of that organic carbon. And so let's compare nitrifiers and heterotrophs directly, because really understanding the distinction between these groups of organisms is going to be fundamental in understanding the differences between nitrification and typical BOD reduction. Looking at this graph, we really can visually see that the growth rate or number of cell divisions for heterotrophic bacteria is going to be significantly higher in comparison to nitrifiers. This essentially boils down to the fact these groups are utilizing those two different food sources as we discussed earlier. We're using low energy producing inorganic nitrogen for nitrifiers and high energy producing organic carbon for heterotrophs. This difference really not only affects the growth rate, but as we see later, it's also going to affect the recovery rate and ability to recover after upset for these two different groups of organisms. All right, and so now let's compare nitrifiers and heterotrophs, but look specifically within a wastewater system. Wastewater treatment systems contain a combination of these two groupings of organisms, and as I stated before, nitrifiers have a much slower growth rate due to utilizing that nitrogen as their primary energy substrate. And so when we take a look at the doubling rate of nitrifiers, we see that the doubling rate is anywhere between 22 and 48 hours. In comparison, we can take a look at heterotrophs, and we can see that heterotrophs have a doubling rate of between 20 and 30 minutes. So this really is quite the difference. This difference not only leads to a faster growth rate for heterotrophic bacteria, but also tends to help achieve a higher overall proportional population within a wastewater treatment system. As you can see, that heterotrophs take up over around 90% of total the bacteria population within a typical wastewater plant, whereas nitrifiers are anywhere between 4 to 6% of that total bacterial population. So it's really going to be dominated by heterotrophic bacteria. And so as I talked about, the growth rate, the ability to recover, I want to now talk a little bit about Aquafix's nitrification and ammonia removal product, as it directly relates to that conversation of the difference between nitrifiers and heterotrophs and their ability to recover and grow after an upset within a system. Aquafix's nitrification product is VitaStim Dynamic Duo, and it's what we use to reseed nitrifying bacteria within a wastewater treatment plant and either recover or boost nitrification and ammonia removal. We produce laboratory cultures of nitrifying bacteria in-house and prioritize diversity and adaptability to different environmental conditions in order to have the highest success rate of establishment across varied field applications. Our nitrifying bacteria are also produced through a batch culture process and are included with environmental cofactors and micronutrients to support a quick population growth and startup and establishment within a wastewater treatment system. Dynamic Duo, as its name suggests, is a two-part product, and I'll show you a little bit more about each part now. So the first part of Dynamic Duo is going to be VitaStim nitrifiers. VitaStim nitrifiers is made up of our autotrophic nitrifiers, and they follow that nitrification pathway that we looked at in the beginning of today's webinar. We're oxidizing ammonia to nitrite to nitrate. As I move on here, we're going to look at the second part of the Dynamic Duo product, and that's going to be ammonia assimilators. So these are the bacteria that take up nitrogen from organic sources and are heterotrophic and are able to assimilate that organic nitrogen directly into their cellular structure. It's just another way of lowering the ammonia in a treatment plant, and it also works at a very low DO, which is pretty handy for some systems who tend to struggle with maintaining sufficient oxygen levels. And although this is really a secondary process for ammonia removal compared to that nitrification pathway, together it's just two separate pathways and two different ways of getting the ammonia and nitrogen levels brought down within a plant. All right, so after covering VitaStim Dynamic Duo, I want to now take a look at what that process of ammonia to uptake and removal looks like within our lab and on a graph. So this graph shows a recent run of our ammonia uptake testing using a dose of our VitaStim Dynamic Duo product in 100 milliliters of nitrifier media. This testing was run on nitrifiers that were stored in our fridge for 6 months, and 50 parts per million of ammonia were present within the flask at the start of testing, as you can see at the start of the red line on the graph. So the ammonia removal process is going to work just like this, where we initially see ammonia levels in the red at around 50 parts per million begin to decrease as nitrifiers are able to oxidize that ammonia into nitrite, that first step of the nitrification pathway. As ammonia is oxidized into nitrate, we can see in orange our nitrite levels begin to increase within our treatment system here. As nitrite begins to build up and increase, we can then see that that nitrite starts to be oxidized to our final product of nitrate in green, and slowly we end up getting more nitrate then. And so at the end of this 15 days here, we can see that we have 50 parts per million of ammonia completely reduced to zero. We see that we have our nitrite slowly creep up and then eventually get reduced as nitrate is produced to zero. And then we see we end up with around 50 to 60 parts per million of nitrate at the end of this testing. And this really is just to show you the process of ammonia removal and the nitrification pathway. It's important to note that the oxidation of nitrite appears to be slower in comparison to that initial oxidization of ammonia in red, and we're going to investigate why this is occurring later in today's presentation. Just stay tuned. So the previous slide was showing ammonia removal within a completely controlled environment within our laboratory, but unfortunately, nitrifiers in the environment are a little bit more difficult and unpredictable to deal with, I would say. First, nitrifiers are present almost everywhere, as I said, in the environment, from soil to the air to the water. However, nitrifiers are also very sensitive to environmental changes around them. And this is where it becomes difficult to maintain a healthy population within a wastewater treatment system, especially if your system or influent is experiencing changes. Finally, there is really going to be a stark difference when we talk about these environmental or system changes in regard to chronic or acute issues. Acute issues provide hope for recovery after the conditions return to normal, whereas chronic issues with environmental conditions can make it exceedingly difficult to maintain consistent and long-term nitrification. Well, there's a lot of things that can affect nitrifiers, and there's a reason to be able to understand why all these environmental parameters are affecting nitrifiers and what we can do to help. So we'll move on now and we'll discuss some of these environmental parameters a little more closely. So the first environmental condition I would like to talk about would be one of those treatment system conditions, and that is retention time within your wastewater system. And so When we're talking about retention time, this is just going to be to get a perspective of what may be going on within your activated sludge and how that can relate to overall ability to be able to have efficient nitrification occurring within your system. And so some prerequisites for nitrification are healthy flock-forming bacteria and sufficient and good BOD removal before that nitrification can occur. Healthy flocks mean your sludge age is really not going to be too young or too old, because nitrification isn't going to work so good at either end of the spectrum of sludge age. If your sludge age is too young, you may have more BOD coming into your plant than your bacteria can remove in a good time frame. And this means that nitrifiers can't get enough oxygen to start oxidizing ammonia and begin that nitrification pathway. The flock formers at a young sludge age will use up all that oxygen as fast as they can as they start reducing that BOD, and can really outcompete the nitrifiers for that oxygen source. And this is going to lead to inefficient nitrification from occurring. If your sludge age is on the other end of the spectrum and too old, you may see poor BOD removal or the disintegration of flock and the breakup of flock as bacteria inside it die. Having flock break up can really harm nitrifying bacteria. As I said, that they are inhabiting the outer edges of these flocks and are really going to be a little bit more fragile to changes in conditions such as flock breaking up. Old sludge can also favor the growth of nocardioforms or other foaming filaments, and foam and scum can really reduce on the top surface of your basin, can reduce the amount of oxygen diffusion from the surface of the basin into the rest of the water. And this coverage can be a significant reduction in oxygen within your mixed liquor, and then that can also lead to lower nitrification efficiency. So really, it's tough, but both ends of the spectrum of sludge age are not going to be really viable for nitrification to be efficient and occur. And you really want to find out what that perfect happy medium is for your specific treatment facility. So here's a visual representation of flock formation as it relates to sludge age. And as we see, we go from a young sludge with free bacteria to bacteria producing EPS and starting to flocculate to healthy, fully formed flock. You're going to want to see a similar image to the far right, where you have somewhat condensed and irregular shaped flock. And this is going to happen at that correct sludge age or retention time for your specific wastewater system. And this is where nitrification will be optimized. Moving on, I want to briefly discuss nitrite lock. And this can really be a limiting factor on your overall nitrification efficiency. As I stated before, the oxidization of nitrite to nitrate, the second step in nitrification, is going to be the slower and rate limiting step of the reaction as a whole. Because of this discrepancy, nitrite oxidizing bacteria are often not able to keep up with ammonia oxidizing bacteria, causing a buildup of nitrite within the system. A buildup of nitrite can temporarily limit ammonia oxidization from occurring and can lead to elevated effluent nitrite and ammonia levels leaving a wastewater treatment system. This can really often occur, and we see this often in systems with large inputs or dumps of ammonia coming into your system at once, leading to this buildup and lock of nitrite levels. So moving on, I would now like to discuss temperature. And temperature is a really important one, as I saw from the poll that a lot of your treatment systems have been affected by temperature changes, causing a stress on your nitrifying bacteria. As I said, here in Madison, it is very cold this morning, and obviously those nitrifiers within the Madison Municipal Wastewater District are most likely having a tougher time nitrifying ammonia compared to a nice summer day. So when I'm talking about temperature, I want to first focus on rapid temperature changes and how they can have a significant adverse effect on nitrification. And that's really because nitrifiers are unable to adjust quickly due to their slow growth rates. Most bacteria in municipal wastewater, including nitrifiers, will have a slower metabolism and slower growth rate as temperature decreases. Nitrifiers prefer a temperature range between around 15 to 30 degrees Celsius, and that's 59 to 86 degrees Fahrenheit. And they have trouble growing fast enough to maintain a population when you get below that 15 degrees Celsius. And sometimes have problem with low dissolved oxygen levels actually when you're above that 30 degrees Celsius range, as oxygen solubility decreases at those higher temperatures. In the graph on the right, we tested ammonia uptake for nitrifiers in different temperature chambers, 23 degrees Celsius and 7 degrees Celsius in the blue. And as you can see, the rate of ammonia removal was significantly slowed at a colder temperature. And so I guess actually I want to say one last thing about temperature. Sorry about that. But I want to say that Some populations of nitrifiers may be adjusted to a lower or higher temperature over time, as nitrifiers still do have the ability to acclimate to consistent temperatures outside of their optimal range. However, really prolonged cold or these rapid fluctuations, that's what's going to be really detrimental to a nitrifier population as a whole, as those rapid fluctuations, nitrifiers just aren't unable to adapt that quickly. And so I'd like to move on now from temperature, and I'd like to discuss dissolved oxygen levels as it relates to nitrification. Obviously, nitrifiers need oxygen to perform nitrification, and nitrifiers perform optimally within a wastewater treatment system that has dissolved oxygen levels between around 2 to 4 parts per million. This range of DO provides sufficient levels of oxygen not only for BOD removal to occur, but also nitrification and ammonia removal to occur. The top picture on the right is going to be an example of what good DO looks like in wastewater flocks. We look at these flocks under phase contrast using the 40x objective with no stain in order to get a sense of what dissolved oxygen levels look like within flock. The picture on the bottom right is going to be an example of what low DO in flocks look like. It's important to remember that nitrifiers need this oxygen to perform nitrification, so it's important to have a consistent amount of DO within your system to allow nitrification to occur, not only within the water, but also within flock. And so now regarding pH, I want to talk a little bit here because nitrifying bacteria are generally working best in a pH range of around 6.2 to 7.9. And the closer to around 7.2 they are, really the happier and more efficient your nitrifier population is going to be. The ammonia oxidization process does produce nitric acid as a byproduct, and too much acid accumulation within your system can drop the pH down. And as the pH gets dropped down, this can inhibit further ammonia oxidization. Conversely, at a higher pH, ammonia oxidizing bacteria will either be dormant or may die at these pHs greater than around 8.5. And also, when you start getting increasing pH of past 8.5, you really end up getting a conversion of ammonium to ammonia, as you can see on the graph. At high levels, either one of these compounds can be toxic to your nitrifying bacteria, but ammonia, the NH3 here, is going to be toxic at lower concentrations in comparison to ammonium. And this is why a higher pH is often more dangerous for a nitrifier population to endure. So because of this, really remaining within an optimal pH range is going to be best for your nitrifying bacteria population. And also, when you're talking about pH, pH fluctuations are really going to be unlikely to occur in most systems, but especially in systems with sufficient alkalinity. And regarding alkalinity here, it's going to be defined as the buffering capacity of a water body and is essential to control pH levels within the optimal range for nitrification. Carbonates and hydroxides are major contributors to a system's alkalinity level overall. And interestingly, nitrifiers primarily use inorganic carbon, such as carbonates, carbon dioxide, and bicarbonates, as their primary inorganic carbon source for cellular components and conversion into inorganic or organic carbon. These compounds that make up alkalinity also help sustain consistent pH values within a treatment plant, making it all the more important to maintain sufficient alkalinity within your plant. As I said before, these nitrifiers produce acid during the process of nitrification and can drop your pH down. Typically, alkalinity around 100 parts per million is optimal for nitrifier function. However, this value is flexible for specific treatment systems that work best for you. So for plants that are struggling with low alkalinity levels or pH swings during nitrification, Boost and Lock is a product offered from Aquafix that can be a solution for you and for your nitrifier population. Boost and Lock is a combination of carbonates and hydroxides that can sufficiently increase alkalinity levels within your plant. And Boost and Lock is not only able to effectively really neutralize the production of nitric acid during nitrification, but it is also able to act as an inorganic carbon source for nitrifiers as well. The combination of ingredients in Boost and Lock can provide a superior stabilization product in comparison to the addition of any ingredient just on its own, such as being more soluble than just lime on its own. And so I want to move on to our last environmental or system parameter that can affect nitrifiers for today's presentation. And one of the most important parameters, I would say, as I would like to discuss toxicity and how toxic compounds within a treatment system's influent can adversely affect a nitrifier population. Upstream sources, often industrial, can be common sources for toxicity coming downstream into a wastewater treatment system. And nitrifiers are often more susceptible to toxins in comparison to other wastewater heterotrophic bacteria. And this is largely due to their thin cell wall and their slow rate of regrowth after being exposed to toxins. This makes recovery after toxicity exposure all the more difficult for nitrifiers specifically. These biocide cleaners and sanitation compounds can remain present within influent, and exposure to nitrifying bacteria can limit nitrification within a plant. Aquafix has done extensive research on toxicity's impact on nitrification, and our research has primarily revolved around quats or quaternary ammonium compounds. We are also currently running this year some new testing on PAA, or peracetic acid, and that effect on nitrifying bacteria. So I will go here and say some takeaways for this first half of the presentation before we move on. And my first takeaway that I want to highlight is that really nitrifying and heterotrophic bacteria are very different. They both inhabit a separate ecological niche within a wastewater environment. And it's important to remember that they are really, really different when taking into account your heterotrophic bacteria and your nitrifying bacteria. Secondly, nitrifiers do have a delicate relationship with the environment and can be affected by a multitude of environmental parameters or system changes. And because of that, the repopulation of nitrifiers and the ability to maintain efficient nitrification is going to be difficult, and more difficult in a lot of ways than maintaining sufficient BOD removal. And so now I would like to move on here and go through some of that research that I talked about earlier, and really some of the specific research we've been doing at Aquafix on nitrifiers, as well as show some nitrifier application case studies from the field. Starting off here, I'd like to show some in-house data from testing of our nitrification product VitaStim Dynamic Duo in order to accelerate recovery after a toxicity event has occurred within a wastewater treatment mesocosm. As I said, the primary toxin we have analyzed is quat, and that is what we are analyzing in this testing. And really, this data is expanded on, and much more can be seen in the research paper that Derek shared with all of you earlier today in today's webinar. So feel free to read that as well, but I'm going to go through some of the highlights here in today's webinar. So starting off, we wanted to show a baseline for how quat and other toxins can affect BOD and ammonia removal within our wastewater mesocosms in the lab. We can see here at 10 parts per million of quat within our system, we really don't see any adverse effects in regard to either BOD or ammonia reduction. However, as we begin to increase our concentration of quat doses, we see that at around 25 parts per million, we see that the nitrifiers begin to be really affected, and you really start to lose a lot of your ammonia reduction occurring when there are 25 part per million of quat being introduced into our wastewater treatment environments here. It's important to note here that even though nitrification is really starting to be hurt by the presence of quats, we see that BOD removal is really not hurt really too much until you get to a really high concentration of toxicity coming in. So you really want to see here, and looking at this graph, it should show you that nitrifying bacteria are going to be more sensitive to the presence of toxins, and the ability to remove nitrogen is going to be harder under the presence of any toxins coming in. And so this is really one of the primary reasons why nitrification is a more fragile process in comparison to overall BOD removal within not only the lab, but also your plant. So now we can take a look at recovery rates between ammonia removal and BOD removal after a 50 part per million toxic event containing quat. So I first want to point out the control lines in the graph in dark blue and for control COD and light blue for control ammonia. We can see that throughout the experiment, our control COD and ammonia levels in our wastewater mesocosms remained consistent throughout the entire experiment. After toxin exposure on day 0 at 50 parts per million of quat, we can now see effluent COD levels rising to around 165 parts per million in comparison to 40, and we can now see ammonia levels rising to around 20 parts per million from 0. Now, let's take a look at the recovery time differences between COD and ammonia removal after that toxic exposure. So you can see here from these boxes that the recovery to our control COD levels really takes about 2 days after our COD levels spike, after that toxic exposure. We get back to a consistent level around 40 parts per million of COD leaving our system. We can compare that to our ammonia levels, where we see our ammonia immediately spike up to around 20 parts per million on day 1 after that toxic exposure, and we don't really get any ammonia removal truly occurring until day 6, where we start to see ammonia start to be recovered, and we get full ammonia removal recovery and nitrification recovery around day 7. So looking at this in a controlled environment in the lab, we can just see how hard it was to recover from ammonia removal, and it just takes a longer time in comparison to the recovery of BOD removal in response to a toxic shock within your system. And so I want to look now not only at the response for BOD and COD removal, I want to look at how can ammonia removal be helped with the application of VitaStim Dynamic Duo after nitrifiers are affected adversely by exposure to some toxin such as quat. So again, we have a wastewater mesocosm, and we have flasks that were running for 3 days. And during these 3 days, we saw consistent ammonia reduction during this test. On day 3, we had 10 parts per million of quat fed into each wastewater flask. And as you can see from the graph, after toxin exposure, nitrifier populations were damaged and ammonia levels climbed in our wastewater flasks. Now look at day 5. Day 5 is when VitaStim Dynamic Duo was added to half of the reactors for a one-time dose application. VitaStim Dynamic Duo was only added to half of the reactors in order to compare a control recovery of ammonia of nitrification compared to a VitaStim Dynamic Duo boosted recovery of nitrification. What you can see here now is after the application of Dynamic Duo, we can see that ammonia removal really rapidly recovered in our wastewater reactor flask in comparison to the control flask that had to recover without any product being applied at all. So as you can see, this recovery being more efficient, we hope this can show you that VitaStim Dynamic Duo can really help to reestablish your population of nitrifiers, accelerate your recovery in order to have efficient nitrification occurring within your plant, and lower ammonia levels after an upset. So our nitrifying bacteria can help restore nitrification after an acute upset. However, it's still important to remember that difference between acute and chronic environmental issues, as chronic environmental issues can permanently inhibit efficient nitrification from occurring. So really, not only focusing on recovery, but also focusing on what the environmental or system issue is that's causing your nitrification to be inhibited is really important, both parts of that problem. And so you need to make sure that not only is that chronic issue fixed, but that acute issue is done with before efficient nitrification can be re-implemented into your treatment plant. And so I wanted to now circle back briefly just to show our VitaStim Dynamic Duo nitrification product again, after showing some of that in-lab data about nitrification recovery and getting back your efficient ammonia removal. And so our product is based upon our laboratory-produced culture of nitrifying bacteria, as well as those other environmental cofactors and micronutrients that help establish growth quickly within your respective treatment system. So our product, the goal is really to help recover nitrification as well as establish a better ability to organically assimilate nitrogen within your plant after an upset. Our products are typically added at the same time to use both parts simultaneously within your system. And regarding a location, our Dynamic Duo product is typically applied directly to our aeration basins in order to get a quick drop in ammonia and reseed your native nitrifier population and establish whole growth for that population. After 10 days, we can typically have strong results in regard to ammonia removal recovering and nitrifier populations having sustainable growth moving forward, as long as we don't have any chronic environmental issues still being a detriment to a plant's ability to nitrify. We typically begin to see results starting to occur after about 3 days in municipal facilities and about 7 days for industrial facilities. And finally, our Dynamic Duo product cultures are well adapted to really varied environmental conditions. So we really pride in making our product adaptable to various treatment systems out in the field. So now I'd like to move on from that in-house data and talking about Dynamic Duo. And I just want to briefly show and finish off today's presentation with some case studies from the field of plants out there getting upset and losing nitrification, and then having Dynamic Duo be applied, aiding in their recovery. So I first would like to start in the municipal plant in Pennsylvania. This particular plant unfortunately had a large diesel fuel spill upstream, about 7 to around 55-gallon drums worth, and this occurred upstream and flowed into their municipal process. Typically, this plant had about 220 parts per million BOD and about 23 to 32 parts per million ammonia coming into the plant. So really pretty normal for a municipal plant. Temperature was somewhat cold at 13 degrees Celsius during this event, so we were below that optimal range for nitrification to occur. After that initial diesel spill, BOD removal recovered pretty quickly, but nitrification remained limited as effluent ammonia really continued to increase for multiple months after the spill. With temperatures left remaining low below that optimal range, recovery appeared to seem difficult. And so what our solution was here was to begin feeding Dynamic Duo at 1 gallon per SBR starting the first week of January. And even in these cold temperatures, efficient ammonia removal was able to be recovered after a week of treatment, and effluent ammonia levels returned to normal at less than 1 part per million after the application of Dynamic Duo. Moving on, I want to move from a municipal example and show an industrial example here for nitrification recovery. We worked here with a large barge cleaning facility in order to help restore nitrification. This barge cleaning facility had 2 batch reactors in their system, with typically 250 parts per million ammonia coming in and eventually 1 part per million ammonia leaving the second reactor. After a suspected toxic event occurred, effluent ammonia levels had risen to around 25 parts per million. And so our solution here was to start another 10-day treatment of Dynamic Duo being applied at around 3.5 ounces of nitrifiers to the first reactor daily. And after 10 days in this industrial system, we were happy to see that effluent ammonia levels had recovered to 1 part per million, their standard, and a healthier nitrification population was established in this particular treatment system moving forward. And so I want to move on from that industrial example into a meat processing example. And this case study here is going to focus on nitrite lock recovery. So not only was it not just recovering efficient nitrification, but here we wanted to solve nitrite lock from occurring within this meat processing plant. This hog slaughter facility here had an activated sludge process that included a 1.8 million gallon aeration tank. Their waste stream heavily favored ammonia in comparison to BOD, which made things difficult as ammonia levels were around 200 parts per million and influent BOD levels were around 30 to 40 parts per million, so not really typical. Effluent ammonia was typically less than 1 part per million when running efficiently, and one note is that the alkalinity in this plant was typically 120 part per million, but it could swing down as low as 50 part per million. So the issue began when heavier loading led to a rapid drop in pH down to 6, as well as a pump malfunction led to a loss of significant solids from the aeration tank. Both of these issues combined led to ammonia spiking to 5.86 part per million initially. Interestingly, after pausing treatment and allowing time for a natural recovery, effluent ammonia levels did and it eventually returned back to normal levels and ammonia oxidization resumed. However, the operator noticed that nitrite effluent levels continued to increase up to 45 part per million after the initial upset, and thus complete nitrification still wasn't occurring in the plant. Even with ammonia being reduced or being oxidized, we saw that nitrite was left there and was blocking nitrification from fully occurring. So our solution here to solve with nitrite lock was to reestablish the nitrite oxidizing population of nitrifiers through the application of Dynamic Duo. So as I said, when we have those the nitrifiers that we are able to seed within the plants, we are not only helping ammonia oxidizing nitrifiers, but also nitrite oxidizing nitrifiers. So after a 2-gallon initial dose, nitrite levels were rapidly reduced to 7.7 parts per million from that 45. And unfortunately, as with everything out in the field, a cold temperature shift occurred and nitrate levels then eventually rapidly increased back to 32 parts per million after cold temperature shifts occurred. So we continued treatment and continued dosing our VitaStim Dynamic Duo, and we ended up getting a full recovery, and nitrite lock issues were solved. So we were able to establish complete nitrification once again within this meat processing plant. And so after these case studies, I just want to discuss briefly some of our total takeaways from that in-lab research, as well as that work in the field with nitrifiers and nitrification. The first point is that nitrification recovery after upset is difficult and can take an extended period of time. It's going to be much more difficult than recovering your BLD removal in regard to recovering nitrification. And that's where we really pride ourselves in being partners and trying to facilitate that recovery and create a faster recovery and faster track to efficient ammonia removal and nitrification. That's where we come in with VitaStim Dynamic Duo, and this is really all this research. These solutions have been through extensive laboratory and field data that we've collected in trying to have really optimize our ability to recover and help plants recover from upsets in regards to their nitrification. And so with that, I would really like to thank all of you for joining us today. I hope you're able to learn a little bit more about nitrification and wastewater, as well as a little bit more about our solutions for recovering nitrification after an upset. All of our past webinars can be seen at the following link on the Team Aquafix website. And I wanted to take a moment to say that we do offer a certificate for attending today's webinar. And so with that, I'll try and answer some of the questions from the presentation. So Robert asked, is nitrogen harder to remove in winter? Well, I hope that the presentation was able to answer some of that. But yeah, as temperatures get colder, it's going to be really a lot more difficult for any bacteria to be able to metabolize nutrients at that same level as during optimal, hotter conditions. And so cold temperatures really do have a negative effect on nitrification and reduces the ability of nitrifiers to reduce ammonia. So nitrogen is going to be harder to remove in winter. It's not impossible, but it is going to be harder to remove in winter. Benjamin asks, can polar bacteria help achieve better ammonia levels? So I guess I assume you're speaking of bacteria that are more adapted to cold temperatures and more acclimated to a colder environment to help achieve better ammonia levels. Yeah, absolutely. When you have bacteria that are more acclimated to colder temperatures, you're going to be able to achieve better ammonia levels in the winter. It's more a point of, just trying to have your entire nitrifier population be able to try and work together. There's no magic magic bacteria or magic nitrifier that is the perfect cold weather nitrifier, but it's working with a diverse population that can be acclimated over time to colder temperatures in order to work more optimally in reducing nitrogen at those temperatures. So will nitrogen or ammonia levels affect BOD? Nitrogen can affect your BOD measurements. As I said, and those organic molecules do contain lots of nitrogen. So your nitrogen is going to be a part of that BOD as well in those organic proteins, amino acids, and everything else that's going to be registered under your BOD. Thanks, George. James asked, could you help sending samples to you from Canada for lab analysis? Absolutely. James, if you want to reach out to either go to our website or reach out to orders@teamaquafix.com through an email, or feel free to call whenever. We absolutely could analyze any samples for you within our lab, even from Canada. So Thomas asked about nitrite lock here and how do you recover from nitrite lock? So recovering from nitrite lock, a lot of it is time. As I said, those nitrite oxidizing bacteria are just simply not as efficient as ammonia oxidizing bacteria. So time is a first way to recover from nitrite lock. But the best way to recover from nitrite lock is really supporting those, not only the nitrite oxidizing bacteria, but also the ammonia oxidizing bacteria, to be able to have an efficient full nitrification reaction from occurring of going from ammonia all the way to nitrate. So it just, as I showed in that one example in the case study in the field, that in the meat processing plant that was having nitrite lock issues, supporting those nitrite oxidizing bacteria with Dynamic Duo and establishing better growth can help recover from nitrite lock and be able to have your full nitrification process occurring at that point. Anne Marie asked, what about if the DO is 9 parts per million? So if the DO is not in that optimal range of it, we were saying at 2 to 4 parts per million for a typical wastewater plant, it's not going to be the end of the world. A higher DO level should be okay, as long as you still have good floc formation for nitrification to occur and for nitrifying bacteria to remain present and happy in your system. The issue when regard to DO, the more pressing issue is going to be when there is not sufficient DO and oxygen levels in order for nitrification to occur, Anne-Marie, because without that oxygen being present and available for nitrifiers, you're not going to be able to oxidize that ammonia to nitrite and oxidize that nitrite to nitrate. So DO at 9 should be something that is okay in your specific system. We can talk further about that if you would like. But the more pressing issue typically is if DO is actually lower and there's not enough oxygen available. Doug asked, boost and lock available in 1,500 to 2,000 pound super sacks. I can check on that, Doug. I don't have an answer off the top of my head. Our sales team would be able to get an answer to you very quickly on that, but that's something I can. I'll take down your email, Doug, and we can get an answer to you for that, and we can reach out to you if it is available in those super sacks. Richard asked about meth lab toxicity coming into your plant and how that can be, and that's something that absolutely could damage your nitrifier population. Anything that is not going to be your typical waste stream can be a detriment to your nitrifier population, Richard. Our specific research was on quaternary amines and those quat compounds, but we are also researching PAA now for toxicity. But there's a ton of compounds and different chemicals that could come into your waste stream that could produce that could have a toxic effect on your nitrifying population. Terry asked, what would be the dosage rate for 1.5 million gallon SBR? Terry, on the handout on the Dynamic Duo product sheet, we can look quickly at that to see we should have some dose rates there, but it's always good to talk to your sales rep for the best dose rate for your specific treatment system and what you're dealing with. And so we can get that for a 1.5 million gallon. It's not on our typical sheet, so that's something that we could talk about and could get you an answer for a preferred dosage rate for your system and also your situation, as that dosage rate can be flexible based on not only the size of your SBR but also how quickly do we want the nitrification to be reestablished, as well as how much toxicity there has been or what the upset actually is. So we can get a better answer to you, Terry, on that. I'll take that note down and we can discuss that and can talk further. Matthew's asking, can you over nitrify? I wouldn't say you can over nitrify. One thing that can be an issue is if you have a slug load of ammonia come in, you can have your ammonia oxidizers over nitrify. You have a buildup of nitrite and you get nitrite lock in that case, Matthew. Also, if you have a very old sludge age, there's not much alkalinity to be able to control those pH swings in the production of nitric acid from nitrifiers during nitrification, then you can over nitrify and have a pH swing, and that can affect your system as well. But typically but other than that, there wouldn't be much for over nitrifying. And Doug, you asked about our Aquafix products, WTAs in Michigan, and if they're approved for Michigan use. Most products would be. One of our sales reps can reach out to on that to get an official answer of which product you're looking for, Doug, and if that could be approved and used in Michigan. Most would be absolutely yes, but we can get a specific answer to you, Doug, on that, on which product you're thinking of and what else you need to know for permitting and approval in Michigan. I believe I got to most of the questions. I just want to thank everyone again for staying through with this presentation and joining for today's webinar. Feel free to reach out if you have any questions after today's presentation about nitrification and possible solutions. But again, I just want to say thank you again for joining us today. I hope all of you have a great day and learn something from today's presentation.